Everyone, and thank you for joining. I'm Matt Roth with Baylor College of Medicine, and I welcome you to today's presentation as part of the Extracellular RNA Communication Consortium webinar series. The ERCC is an NIH Common Fund program that works to advance the science and research of extracellular RNA. Stage one of the consortium started in 2013, and the second stage began in the fall of 2019. And through this webinar series, the consortium hosts monthly presentations of a variety of research topics related to xRNA. I'm very pleased to introduce today's speaker, Dr. Michael Graner, Associate Professor in the Department of Neurosurgery at the University of Colorado Anschutz Medical Campus, where he serves as Research Director and Steering Committee Chair. The title of Dr. Graner's presentation is Viral-like Impacts of Glioma Extracellular Vesicles on Normal Brain Cells. Michael?
Thanks, Matt. Thanks for inviting me, and thank all of you for attending. You'll notice I've co-opted the ERCC symbol here because it's like cheering for whatever team wins the Super Bowl after the fact, and you get to buy the T-shirt then. So thanks for letting me be a part of this. You're going to find there's really very little about RNA, extracellular RNA directly involved. So I feel this is I'm a little bit out of place, but Matt dragged me into this, so we're good. Okay, disclosures, really nothing. You're going to see some cats and dogs, and this is my trademark, cats and dogs showing up in my talks. So try to bear with that. All right, so the problem we work on is brain tumors, and particularly the worst kind are things called glioblastomas. These are the most common of the high grade of the astrocytomas, period, and they're the most deadly. Patients survive usually less than 15 months, and pretty much no one gets out alive. It's almost universally fatal. What's shown over here is a slice through someone's brain, so obviously things went badly for this person who had a glioblastoma, and it shows you all the features of them that are why it's so nasty. Among them, proliferative. This has covered an extraordinary area of the brain. It's invasive. It's crossed the blood-brain barrier, or the corpus callosum, which is kind of a surgical no-go for most patients. In this case, vascular. It's a bloody mess with a hypoxic core, or sort of this messy, gooey core here. The rest of it is highly viable, spread out across the brain like that. Even with the vascularity, there are extraordinary areas of hypoxia in here, which leads to very heterogeneous cell types in terms of their genomic makeup, their expression makeup, and how they respond to various stresses. And that makes them extremely resistant to therapy, which is why we are very seldom successful in doing much for these patients. Our therapies have been the same thing for the past 30 to 50 years. Maximal surgical resection, followed by forms of radiation, usually combined with chemotherapy, in this case, temozolomide. And we end up falling back on maybe experimental therapies to try to help these patients. And these are not doing spectacularly either, despite our best efforts. So we need to have a better understanding of the biology of these things to try to get a better grasp on how to treat them. Other obvious therapeutic difficulties, because what's in the brain, nothing good can really go on if you have to start bashing on the brain to try to make your therapeutics work. The blood-brain barrier is another formidable means of preventing things getting in. This is a great evolutionary mechanism to not let noxious compounds enter into your brain so you don't fry yourself. But on the other hand, when we try to get those noxious compounds into the brain because we're trying to treat a tumor, it can make it very difficult. It's considered an immune privileged site, which means it's kind of a specialized area as far as immune responses go. And they tend to be dampened down so that if you do get an infection in the nervous tissue, you don't end up having yourself fry with an inflammatory response. So it's good in some respects, but once again battles off the damage we try to do to the tumor with the immune responses. And these brain tumors are extraordinarily immune suppressive as well. In addition, anything we do in the brain can cause further effects if you have CNS difficulties, such as exacerbating a stroke potential. We have a number of patients who suffer strokes either during surgery, after surgery, or following treatment because the radiation sort of breaks down the brain itself. So there's really nothing good about these, pretty much at all. And our ability to treat them is very, very limited, let's put it that way. And I don't think I'll have much of an argument from anybody on this is a bad disease. So I'm not going to spend virtually any time talking about exosomes, extracellular vesicles in terms of how they're generated, what goes in them, all of these things you already know. Just want to point out that this is a sort of, when you submit something like this to a journal and a reviewer basically asks, has the writer had a stroke? No, I hadn't. I'm just not that good at this. And they had to explain to the reviewer that somebody else would fix the problem, and they did. So this is what things look like when you've got a true artist doing the job for you. Something we tend to make these cartoon drawings that are nice little spherical vesicles, things that are round, that have a nice double membrane, to lipid bilayer on it. And work done from Jan Lovell's lab has shown that's probably a little short-sighted. There are a lot of different vesicle shapes, sizes, and some of the drawings of them are down here. They're pretty amazing. So bear in mind that what we're calling these extracellular vesicles is maybe more complicated than we even realize that they are, and we know that they're pretty complicated. We tried to do some cryo-EM ourselves. It didn't turn out quite so well. I need to ask Jan how he got that to work. So our idea, and I think your ideas as well, is that these vesicles are sort of God's gift to communication biologically. Cell types pass them to other cell types. They can be passed to different tissues. They can be passed throughout an entire organism if it's able to enter into, say, circulation of any sort or any way of biodistributing itself about. So this is sort of where I believe we're all on the same page on that. So when we began this work, we were originally just bashing vesicles a lot, trying to find out what was in them, and that we kept finding, in terms of proteomics, many of the same things over and over again. So we decided to ask a different question, is how do the vesicles that are released, say, from a tumor affect recipient cells, things that are not the tumor, or could be the tumor itself? And we're not signaling people, but we ended up following signaling pathways, which was not my idea, but it's kind of the way that the data turned out. So just to point out a couple of things we've seen is that they'll make tumor cells crazier, and extracellular vesicles from brain tumors will turn T cells into teenagers, and I'll explain what I mean by that in a second. So when we take GBM cells, these are grown as a neurosphere, and they spit vesicles out, and we put them back on tumor cells, things happen. The tumor cells will proliferate, they undergo some metabolic changes, which often leads to resistance to chemical treatments that we use, such as temozolomide. They'll invade and migrate at much more proficiently. They will chase the vesicles in terms of migrating across membranes, and the signaling pathways become more engaged, and it's kind of a bedlam sort of formation. We've done this with stressed vesicles, those that came from stressed. If we stress these cells and then take those stressed vesicles, so to speak, and put them on the other cells, this is exacerbated pretty dramatically. We call them sexosomes. That hasn't caught on, and no one's allowed me to trademark that, but that's sort of the story behind stressed vesicles actually causing more of a problem and causing more, for instance, of a tumor resistance phenotype. But in this case, we moved on to looking at T cells for a while. The idea being that if we exposed the glioblastoma EVs to T cells, what would happen to them? Most people thought they'd die, and in our hands, they didn't. What we did end up seeing was wildly dysregulated signaling, and the cells would proliferate, which is usually a sign of activation. But if we activated them and then treated them with the vesicles, they lost that activation status. They were no longer spewing cytokines, and they wouldn't kill target cells. And another thing that happens is that their motility was reduced, which is unusual. We've always seen cells chase extracellular vesicles to migrate, and this actually reduced their motility, which is sort of a unique form of immune suppression if you kind of think about it that way. If the T cells cannot enter the solid tumor, they're not going to have any impact on it. So this is why I refer to them as being teenagers. They're living in your basement, they're eating you out of house and home, and they're not doing a damn thing, and so are these cats. So we asked the further question in terms of what would be cells that would interact with a tumor, a brain tumor, and these are going to be astrocytes. And the data I'm going to show you today are going to try to imply that the astrocytes become gangsta wannabes. So they're going to be these tough characters, become somewhat enforcers for the tumor in this case. But we did not make any money from the profits of this movie, by the way. So, what are astrocytes? This is something that, as somebody who's in the Department of Neurosurgery, I'm probably one of the world's worst neuroscientists. I thought the brain is made up of two things, neurons and brain tumors. Well, it turns out there's a whole bunch of cells, and some of them, the vast majority of those cells, perhaps up to 50% of the brain mass, are actually astrocytes. And these are considered what they would call the glue of the brain. So they're glial lineage cells, and their main job is to support, and by support we mean various things, structurally, metabolically, and in paracrine fashion, in particular for neurons and for the vasculature. So this is kind of their job is to be the cells there to help the other cells do their things. And neurons are particularly needy cells, so they need a lot of support from these. So these would be considered, I think, the most closely linked cells to the tumor microenvironment in this case. We don't have the sorts of cancer-associated fibroblasts you might find in other tumor types, and the stroma is going to be somewhat different. So these are the cells that are probably going to be the most impacted by the tumor and potentially the most impact on it if there's talk back and forth. So just to show you what astrocytes look like in other people's really great microscopy, here we see the astrocytes interacting with blood vessels, and they're forming metabolic units as well as working with things called pericytes. Let's make sure I said that correctly. With the capillary epithelium, this forms the blood-brain barrier. The interactions with neurons are both structural and, in this case, metabolic. The glutamine-glutamate pathways that are necessary to keep the neuron happy. So that's shown here in terms of all the interactions they have. So they're densely populated in the brain, and they have very important support in many different aspects of that. So we took about the simplest experiments you could possibly do, and I was exposing astrocytes to brain tumor EVs. So we take GBM cells, and once again, these are grown as these weirdo neurospheres. These have a stem cell-like characteristic. Collect the vesicles from them and throw them, literally just incubate them with normal human astrocytes, and then ask what happens. And in a paper we put out a while back, the astrocytes release what I call the cytokine chicken soup. They undergo a form of crazed signaling, and then they do things they shouldn't, perhaps, such as migrate and then grow inappropriately. And those are sort of the three big take-home messages from that. And I'll go over a bit of those data in here just to set things up. I realize we're now required by law to show our separation methodologies for EVs. And what we end up doing is mostly centrifugation, high-speed centrifugation, and then characterization with exocheck arrays, electron microscopy. This is ugly, I admit that. And then, of course, the nanocyte type of stuff. And we can argue about this all you want. I'm not going to argue about it. I'm just going to refuse to talk about it. But let's call these things, shall we, high-speed extracellular fractions, if that's going to be a better way to describe it. Seen over here are the cell types we've worked with. These are actually in neurospheres. So these bars are 100 microns. These are wampum clusters of cells that are growing together. And I want to show you a little more about the G17s up close. Don't worry about the names. Just in this particular talk, it's going to be mostly about something the F38s and the G17s, and you don't need to get hung up on those. These form weird little tendrils like this when they're in culture. So while they're sphery and they can float, they tend to attach fairly tightly. We actually have to blast them off with media to knock them loose, as opposed to just being able to pull them off carefully from the plate. So I think the different cell characteristics lead to different vesicle characteristics, which lead to different outcomes when we expose astrocytes to those vesicles. So one of the first experiments we did was to actually ask if astrocytes would migrate. To be honest, the first experiment we did was to throw vesicles on astrocytes and ask, does it kill them? And it didn't. So that was my first thought, was that, well, brain tumors don't really need these astrocytes in their way. They'll throw the EVs out like hand grenades, blow them out of the space, and then just fill in. Turns out that's not the case. So once we realized the astrocytes were alive, we did scratch and sniff assays. These are wound filling assays, and you can see that if you incubate the astrocytes after cutting a hole in their paths, if you incubate them with GBM vesicles, they will fill that path faster than if you hadn't done that. This kind of suggests that the tumors are sort of calling out to the vesicles and calling out to the astrocytes, meaning they might come to or they might come towards the tumor as opposed to being kicked away from it, as was my first sort of suspicion. Another thing they did was they spewed an extraordinary cytokine array, as I kind of mentioned this, and you can see, you don't need to know what these are and you can't read any of it. These are just lists of things in the extracellular space that might be of interest to brain tumor people. And we did this over time, so at 0 and then 4 hours and then 24 hours. So we see there's a massive change over this length of time. And this is, everything here is hot, let's put it that way. So there's a strange cytokine soup, and there's a couple of players that are involved in, say, extracellular matrix remodeling. I'll talk maybe a little more about them later. We took the ups and downs of those expression patterns basically and chunked it into Ingenuity pathway analysis. And we asked what happened, what does it tell us? And we see inflammatory response in cancer as two of the top diseases and disorders. And then other aspects of this, we always see things of cell signaling and interaction, cell movement, cell growth and proliferation. And you'll see this again and again and again. And I'm already tired of it. You'll get tired of it by the time the talk is over. So if we ask about these, what does this actually mean? In particular, the term cell growth and proliferation. If we take the GBM cells, take the vesicles, throw them on astrocytes, collect that media from the astrocytes, and then say throw it back on the tumor cells, what happens to them? They actually grow faster, and that's saying a lot because these tumor cells tend to grow pretty fast in culture anyway. So it's as if the extracellular media, the conditioned medium from the astrocytes that have been treated with the tumor exosomes, they're now making a growth medium, a more effective growth medium for the tumor, suggesting that they can actually promote tumor growth. And while we're talking about cytokine soup for the soul, this is Honduran seafood soup from Berta's in San Diego. If you're ever there, have Antonio de Mayo take you to that. This is a bucket list item, trust me. When we ask questions about what sort of interactomes do we see in this, and these stumbled out of that IPA analysis, and we notice these two major players signaling molecules at these hubs, leading to had to look at signaling pathways. So we did that, and again, taking the GBM cells, the vesicles from them, throw them on the astrocytes, and then asked what sort of phospho signaling changes might be occurring, what sort of phosphorylation changes are occurring that are presumably leading to signaling changes. And you can see over time, we end up with, and this is actually a long time, excuse me, I was disturbed how long this took, but there it was. We see this almost a tumor signature, shall we say, of the signaling patterns that are going on here, which was kind of disturbing. And of course, we threw that back into IPA, and what we came out of it was cancer. Look at that. The idea being that the astrocytes may think they're cancer cells at this point. I didn't want to believe that, but that might be the deal. Cell survival is in there, and changes in tumor morphology, which would be implicated in the idea that the astrocytes can migrate and then develop these signaling pathways. And again, you see this thing over and over again. So we actually asked the question, can we put them in soft auger and what will happen? It wasn't spectacular, but they didn't die, let's put it that way. So if we treated the astrocytes with tumor vesicles, they were able to survive and even proliferate a little bit in soft auger, which is suggestive of a transformed phenotype. This was terribly frightening to me because it sort of implies that the astrocytes may take on sufficient phenotypic characteristics of the tumor that they basically are the tumor. In a recurrent tumor situation where we go in surgically, remove the tumor, treat the patient with all sorts of stuff, and we suspect that the remnant tumor cells we couldn't find or couldn't remove surgically and couldn't kill with our therapies are going to be the ones that cause the tumor to recur. This suggests that the astrocytes may be more involved in that than I really would like to believe, but there are the data. So we've done a lot of work with various types of arrays, and we decided to take a broader swing at things and essentially a transcriptomic work. So from this, we'll have three populations of cells. We'll have astrocytes just sitting there in the dish, astrocytes treated with glioma extracellular vesicles from this cell line or from that cell line. And as controls, we use astrocytes incubated with normal cell EVs. And by normal cell, we have a form of epithelial cells, which is a long story and interesting, but we use those. We've also used astrocyte extracellular vesicles themselves, and we've moved recently to HEKs, EVs, because we can get a lot of them. Astrocytes aren't very good at producing these. So the idea was throw vesicles on, wait 24 hours, obtain RNA from all of this, and do transcriptomics. The whole point of doing transcriptomics, of course, is to get these volcano plots. So you have to wave your hands when you do that. But what we can see here is that there's substantial changes that go on when astrocytes are treated with EVs from tumor cell lines. So this is not unexpected, but a little more than I thought it would be. I was kind of surprised at how broad a pattern this was and how extensive a pattern it was. So we had started to work with some bioinformaticists. I hired one of my cats, and this is just not working out very well. So we instead put all these data back into Ingenuity pathway analysis. And this is from F38. So this is one particular brain tumor type. It's a high-grade glioma. Both of the things we're working with are high-grade gliomas. And we see from the outcomes of this, we see interferon signaling very high as a canonical pathway, interferon-related materials as regulators, and in the disease and biofunctions, we see essentially an antimicrobial response, kind of an antiviral response, along with the inflammation, and then these things which seem to always show up in terms of the molecular and cellular functions. If we look at the molecules that seem to be upregulated, the RNAs that were upregulated, every one of these is actually something involved in either making interferon or responding to interferon. So this is oligodenolate synthase 1, 2, and 3, various interferon-related factors. And we did some Western blots on that to show that indeed, when we treat astrocytes with tumor EVs, they will upregulate OAS 1, 2, and 3 to some extent. Not much on this one. MDA5 and RIG-I, these are players intracellularly that respond essentially to pathogenic issues. So they're kind of an innate intracellular immune responder. And I'll explain a little bit more about them later. We see that RIG-I does indeed go up and these antibodies suck. We also see the increase in GFAP, glial-fibrillary acidic protein. This is a sort of a marker of activated astrocytes, angry astrocytes that are going to be essentially forming what's called gliosis, wherever this damage could occur inside the tumor or inside the brain. Now we expect this, and we know this occurs when brain tumors grow into this space. But what we're looking at here is just a response from the vesicles themselves, not the tumor actually encountering the astrocytes, but literally just the vesicles being thrown on them. No tumor is physically involved in this. So keep that in mind as we go along. The interferon signaling was something we never really looked into because most of the time we only looked at gamma interferon, but all of these downstream RNAs and presumably proteins that seem to be upregulated also fell into the other interferon pathways, such as the pathways 1, 2, and 3 interferon signaling. So reflecting back on history, Flash Gordon is credited with discovering interferon and treated a patient with it in the 1960s, may be relevant these days in our virus-infested world. But checking on the interferons, we probe for a variety of them. I don't even know what interferon omega is, for instance, but in every case, the brain tumor vesicles seem to promote release of these interferons from the astrocytes. So it turns out that was not a complete fake story that IPA just told us. So there it was in our faces. So what does this kind of mean? Well, it seems as if the recipient cells, in this case astrocytes, when bombarded by tumor extracellular vesicles, feel like this is a viral infection. And here's how that works. There's that OAS1 oligodenolate synthase. There's 1, 2, and 3. It converts ATP into 5' oligodenolate. This activates RNase L, and so our OAS1 and 2, 3 got turned on by double-stranded RNA, presumably. This is just one story, which in this case we presume came from a virus. Now the RNase L will cleave that double-stranded RNA into smaller pieces, which are then recognized by RIG-I and MDA5. And the downstream activation effect of that is to make interferons. So this is where those stories tie together. Once again, to show you those Western blots, OAS 1, 2, and 3 are upregulated, RIG-I is up, and the activation of the astrocytes themselves. So we're now facing what kind of looks like a viral infection from these cells, and this is the person who pointed this out to me is my hero, Fatih Kassanci. So I want to thank him for explaining this to me and outlining this paradigm. we then, of course, this is not the full, we always want to do more than just one cell line in case it turns out that that was not the same. So we did this with G17.1 as well. My hope was that we would see very similar outputs. We'd have very parallel stories to tell so that there wouldn't be, we could just keep doing the same experiments over and over again. And of course, that's not the way it worked. And I should have seen that coming. By this time, my bioinformaticists had become a Dark Lord of the Sith. Nothing is helpful here. We moved on and did the same canonical pathway studies, the same IPA type studies, this time on the transcriptomes from the G17 treated, G17EV treated astrocytes. And we see these are quite different from the previous ones we saw. We see that this pops up. There's got to be money to be made in this. I haven't figured that out, but we'll worry about that later. The upstream regulators, things mostly involved in the extracellular environment. And of course, P53 is something that's always a big deal in the brain tumor world, and most other tumors as well. But we get hung up on brain tumors. We see that the top diseases and disorders are, again, cancer and neurological diseases, things that would seem to be a brain tumor. And then all the issues with cell movement and the same things we see over and over again. So the astrocytes feel the effects of the vesicles, but this time it's not the same as from the previous tumor, but some of the outcomes seem to be a little bit similar. Most everything in terms of the upregulated materials were extracellular matrix involved. Periostin, one of the top ones, a link protein for proteoglycans, modifiers of proteoglycans, things like that. So these seem to imply that the extracellular matrix or the extracellular space is going to be another important place to look for things. So we did that again with the media supernatants, asking what secretome comes out of this in terms of an inflammatory response. We see a lot of the same things, but not the same things as we saw from the F3H, for instance. So once again, we're not going to be able to just replicate the story. We have to take it into a different direction and see where these things lead, and just to show you where these things show up on these arrays. And they're probably driving you crazy at this point. And I also want to throw out that once again, the spent media from the astrocytes, the conditioned media, makes tumor cells grow. I gave a portion of this talk at a meeting in Vegas. So this is the seafood buffet at the Flamingo Hilton, another form of things you should probably eat at some point. If we took the overlapping networks, the interactomes, and asked what comes out of this if we pile them on top of each other, put it in this radial hairball formation, we see ERK1/2 is at the center of things. So we're back to the idea that signaling pathways are probably being stimulated by the vesicles from the outside and responding both inside and outside. So yes, more signaling. I'm kind of sick of it myself. I'll try to keep you guys a little more engaged. So what along with the idea that there's ERK1/2 involved, we thought maybe we can inhibit that and see what happens. So this is a complicated slide. This is what the phosphorylation status of these various molecules looks like in just astrocytes just sitting there. If we treat them with glioma EVs, that changes very dramatically. If we throw in an ERK inhibitor, this thing, which is clinically relevant, it changes that pattern a lot, both up and down. Also has some effects on the astrocytes. So there's a lot going on with this slide. And then we, of course, had to just ignore our bioinformaticists at that point, ask what interactors are in that array, so to speak. And here's what we see surrounding ERK, all these things that are touching or talking to it or talking back to, getting talked back to, shall we say. So when I pointed this out, I want to go back one. So if we treat the astrocytes with an ERK inhibitor along with the vesicles, we see that the ERK phosphorylation status does go down, apparently inactivated. That's good. That was what was supposed to happen. A number of other things go down as well, their phosphorylation status decreases. And these can have pleiotropic effects, which we'll see later on. Some things actually go up. So it's not a universal phenomenon that if you take ERK out of this, or if you inhibit ERK in this situation, everybody's inhibited. Some may be, some may not be. These were ones that showed up in the interactant. We don't need to care about that. So going back to some of the stuff that gets released in the context of ERK1/2 inhibition, what happens to periostin? Well, actually nothing. So that was one that periostin is indeed spit out of these cells, not at terribly high levels, but it's significantly spit out. If we treat with an ERK inhibitor, treat the astrocytes with an ERK inhibitor along with the vesicles, it doesn't really change much. It does change TNF alpha release. This goes down quite significantly. Oddly enough, IL-6 goes up. So now when you start putting these into what sort of patterns do we see here? What sort of knowledge do we have of the various signaling cascades that are involved? This gets messy as all get out. So we know these are involved in tumor remodeling, invasion, angiogenesis, and factors that they will deal with are, of course, TGF beta, stimulators of angiogenesis, signaling molecules on the inside. So it gets pretty ugly. And this is a nice pathway or sort of diagram drawn from this Lambert paper, where periostin will signal through alpha V beta 3 integrins, trigger ERK, which then triggers an inflammatory response within F kappa B, and you make more periostin. You also make inflammatory cytokines, and then there's a variety of things that can happen in the extracellular space, including collagen deposition, a release of TNF alpha, which then goes back and feeds into the STAT pathways and the P38 pathways. So when we're seeing the changes that go up and down, they don't necessarily fit the pattern of strict ERG inhibition, taking out most of this. So there are clearly bypass mechanisms, including STATs and P38, which weren't changed all that dramatically in the signaling. So I think that the idea of picking on one particular kinase, so to speak, to solve all of our problems just isn't going to happen. And we can only use it in this case to try to dissect what may be going on. And we're still pounding on this, and we've had obviously issues when you can't go to work. You now just sit and twiddle your thumbs and get mad about it. So to change gears a little bit, rather than working with astrocytes and culture, we've also used brain slices. These will come from epilepsy patients. We're able to get these from temporal lobes. I thought that the brain was color-coded like this, so I didn't know why neurosurgeons make so much money. It turns out that's not true. But in any case, virtually everything we get is going to come from a temporal lobe slice. We'll process these into small pieces. We'll culture them, make sure they're okay. They're actually alive. Believe it or not, these things stay alive for literally a couple of weeks, if not longer. But in this case, we'll treat them as if they were the astrocytes. We will throw nothing on them. We will throw normal cell vesicles on them, and we will throw GBM vesicles on them. We'll look at things in microscopy and then measure stuff biochemically down here. So that signaling array, which you're probably sick of this, I'm getting sick of this. You see, once again, things get really hot, at least and this is from a variety of cell types, right? This is an actual piece of tumor, not merely solely astrocytes. So these respond as well in a variety of ways to being exposed to tumor extracellular vesicles. And these are the various sorts of phenomenon that would be associated with this, including something that popped up as viral infection. One of the things we have done was to do some cool microscopies from two photon fluorescence lifetime imaging for these. And one thing we looked at was what is going on with bound versus free NADH, and why do we care? So in this sort of 2D flim, or 2P flim rather, you're able to measure the fluorescence lifetime change between bound NADH and free NADH. So this is the calibration slide. What does that mean? So we're scanning through this chunk of tissue, which is millimeters thick, basically exciting with a wavelength, I think it's 750, and then collecting whatever comes back through a bypass filter, which I believe was 440/80 or something like that. I don't know the details, and I'm not very smart about this. But what you do is you take all these glows and how long they last, essentially, through the entire tissue, piece it back together, and then throw it into this thing called a phasor plot, which is a bunch of math. I have no idea what it means. But what it amounts to is you should see a shift between bound NADH and free NADH. Bound usually refers to it as the concept being that this is probably in the mitochondria, and the cells that are in that state are undergoing oxidative phosphorylation as their major metabolic output. If it shifts over to the free NADH side, that's going to be considered NADH in the cytosol and is more likely related to glycolysis. So that's what we see here. And when you pile all those numbers together, brain slice treated with PBS, not much happens. Brain slice treated with normal cell extracellular vesicles, not too much happens. But with GBM vesicles, there's a significant change in what's called the tau M average, indicative of a glycolytic phenotype that's now being experienced by these cells. So that was kind of a very long and complicated microscopic way to look at this, which you've also seen with cultured astrocytes and pig brain slices. So we went ahead and actually said, is glycolysis actually occurring in these brain chunks when we treat those brain chunks with GBM vesicles? And to make a long story short, what we're looking at is labeled glucose, 13C glucose, taken up and then spit out as labeled lactate. So the carbons come off the glucose and ended up on the lactate throughout that whole system. And we see a significant change or significant amount of it being taken up by the brain slices and being spit out as lactate by the brain slices. This is kind of the definition of glycolysis as a front end and the back end. A chunk of tissue in culture will put out lactate just by sitting there. So that's why we're using the labeled portion to follow the dynamics of it as opposed to just a constant secretion of it. So it looks like glycolysis is indeed occurring in these tumor tissues, or these brain tissues, when they're exposed to tumor vesicles. Another thing we did was using the forms of two-photon microscopy to essentially look at your lawn. So what we see here are nice dense regions of brain slice. These are neurons, presumably. And then if we treat with vesicles, we start seeing holes in that. And if you reconstruct this in sort of a 3D structure, you see a nice lawn, and then the one treated with glioma EVs, the gophers have gotten in, they've eaten paths in it. This is called a forest plot. I might call it a deforestation plot. What it's indicative of is a destruction of the extracellular matrix. So I'm wondering if these two things are tied together. The glycolysis, which is going to put out lactate into the extracellular space, which is going to lower the pH, which could in theory activate cellular proteases. So we kind of took a look at that, measuring very old-fashioned biochemistry with gelatinase. So if we take the brain chunk, treat it with the various treatments, nothing normal exosomes or brain tumor exosomes, and then collect that media 24 hours later, it has a substantial, significant amount of gelatinase activity in it. That's after sitting there for 24 hours. Now, one could ask, is how much of that is due to the vesicles you put on them themselves? Because we know these tend to have matrix metalloprotease type things with them, and there is activity in those vesicles, but there's not nearly as much. So my thought is there may be vesicle activity early on, and then there's triggering later of the tissue, the brain tissue itself, to release its own forms of extracellular matrix degrading enzymes. We tried to look at this in an astrocyte setting to pull out vesicles from that, and I'm not going to spend too much time on this because this really, it looks like it might be working, but the numbers are really bad, so I wouldn't buy too much on that. So what we got is big chewing from the brain tissue itself and not so big chewing from the astrocytes if they're grown in culture in a small dish where we don't have that many cells and can't collect that much media later on. So we are working on this from a time course perspective. We're trying to work on that until we got shut down. Because this, like I said, this is after 24 hours. The events that are happening early are going to be what's fun between distinguishing what's going on down here. So we're hoping to come up with that later. So yeah, we got top people working on that. I got a new bioinformaticist working for me, and this guy from Vegas who's pretty good. So what could we come up with here? So exposing astrocytes to tumor vesicles. This is stuff we've shown before, the growth and migration, the cytokine chicken soup, the craze signaling. And now we believe there's a significant amount of inflammation involved as well, both through any number of pro-inflammatory and actually some anti-inflammatory cytokines. It's a complicated mess, as well as what can go on in terms of eating sugar and eating the matrix from that. The astrocytes themselves seem to consume more glucose and then are able potentially to eat the matrix around it, with the lowering of the pH and the further release of matrix metalloproteases and things like that. So there's a lot of conclusions to make here. The most important one is that, in general, EVs from the glioma are going to be used to benefit the glioma, and it doesn't matter who they impact, whether it's the glioma itself, whether it's invading T cells, or whether it's the neighboring astrocytes. And just to focus on a couple of things, the astrocytes are going to spew things that make the tumor happy in a variety of ways. They will actually engender matrix alterations, and again in multiple ways. And the glycolytic phenotype, particularly in brain tumor tissue, I would argue contributes to that. We're studying that right now. Just yesterday, got a grant shot down on that, so I won't be pursuing it anytime too soon. But that's one of the goals we have in mind. So I kind of mentioned the concept of cancer promoting EVs a while back with um, the idea that the astrocytes treated with the tumor EVs could actually maintain themselves in soft auger. I proposed this originally to the NINDS a long time ago, and they sent people to break my kneecaps because they did not like the idea at all. It wasn't all that long after that Sonia Mello, when she was in Raghu Kaleri's lab, demonstrated that vesicles from breast cancer cells could take essentially breast cancer fibroblast-like cells and literally convert them into tumors. If those cells were injected into skid mice or nude mice or whatever, they would grow tumors. So I may have been crazy, but I wasn't as crazy as they were. But they got the thing published, and I did not do so well as far as getting funding for that. But it just begs the question, what can we do about this? Obviously, inflammation is a big deal. And inflammation control becomes something we need to think about. But that's a double-edged sword. We often will treat these patients with corticosteroids in order to reduce the brain swelling that occurs from the tumor and from the surgery. This also has pretty dramatically bad effects on the lymphocyte populations, as those often drop. So just blunt smacking with inflammation might not be the best way out. Is it conceivable that antivirals may have some play in this, in terms of not from a viral perspective, but from the cell's perspective of how they deal with a virus? Is ERK inhibition useful? And I would argue not, based on what we've seen here, but that still has a long way to go. One thing people would really love to do is prevent EV release from tumor cells in particular. I'm not sure we figured that out, but I know there are groups out there working on it. And in true Vegas fashion, we could just sing about it and just move on from there. So I want to point out one of my top collaborators in this, Tom Anschutz, who gave up with this statement a long time ago. These are not good collaborators. Tom is. These are other good collaborators. These are the people who actually did the work, got us the samples. Jen keeps us out of IRB jail, and as a variety of really hardworking people who've done a great deal of progress in this thing. So I want to thank them, and I want to thank you, and once again, thank you for being the Vegas type people that you must be right now. So thanks, and I guess, I'm not sure how we'll do this, but we'll take questions somehow.
Thank you, Michael. Very nice. So yeah, everyone is muted, but if you have questions, just unmute yourself by clicking on the microphone icon there, and you can ask your question. If you could just identify yourself and your affiliation, that would be appreciated.
That was interesting. This is Janusz speaking from Montreal. Hi, Michael. It was a very sort of dynamic presentation. I really enjoyed it. My question is, you brought up the whole issue of transformation of stromal fibroblasts by cancer cells. And of course, the question then is, whether in your astrocytic system, this was actually tested directly, or is there any indication that prolonged treatment with tumor vesicles will convert or astrocytes to any kind of tumorigenic, bona fide tumorigenic phenotype, which would obviously require acquisition of certain mutations, such as TERT, for example, or any kind of immortality pathway would have to be compromised, and along the lines of other mutations. So do you think that happens in the context of GBM, that astrocytes become engulfed in a deeper sense in the genetic progression process, or do you look at this more like a stromal response or tumor promoting gliosis?
Most of what I've thought of, most of what I'm willing to believe is there's a gliosis phenomenon that ends up with the inflammatory responses in particular, but also in the cytokine, in the chicken soup, so to speak, of feeding the tumor. This is something that the astrocytes have been essentially compromised to do. And I'm obviously familiar with your work involving the short-term, relatively short-term kind of transformation phenomenon that occurs. which doesn't last. And we didn't check that any further than however long it took to do that experiment. The one thing I would say, and you're well aware of this, is that part of our treatment is to essentially soak the brain an alkylating agent and then blast with radiation. So something has to happen to those astrocytes. And it would not shock me that you've got a p53 mutation in there. And if you trigger that enough in terms of a metabolic change and an inflammatory change, you might be able to convert those things to a bona fide tumor. Now, once again, that wasn't believed when I tried to write that grant. I don't think your paper is out at the time, so I didn't get to beat myself over the head with it. But that's kind of where most of what I think is occurring is actually just a supportive environment. The actual changes is going to, like you said, likely would require a genetic manipulation, a genetic mutation that can throw a switch, but I would argue that can happen. I just don't know that we see that. And by the time the tumor recurs, you can't tell what was an astrocyte previously versus a tumor cell by the time you get hold of it. So
I think this is very interesting because, there are some indications that, recipient cells that ingest tumor-derived, vesicles, they show some signs of genetic instability or impaired DNA repair. So,
right.
I mean, are you looking at this kind of thing, or have you seen certain, I don't know, genetic instability features in your astrocytes or mutations occurring or whatever?
We haven't done that, and I've been inspired by looking at your work that you posted a couple days ago to think about that. What we have seen are more, shall we say, metabolic and structurally metabolic changes. There's sort of a dissolution of the astrocyte, or I'm sorry, of the mitochondrial network that occurs when we treat with these. It turns out it's reversible. There's a drug we found that we can throw on there that will sort of reestablish the mitochondrial network, but that would certainly be indicative of this change in phosphor or this change in glycolytic status. That I also insist, without evidence, leads to the extracellular pH drop, which then leads to the ability to clear a bigger and better path. So the astrocytes end up doing that for the tumor. But that's the work we've done with some pretty hardcore microscopy. But like I said, we haven't looked at the potential that these are very unstable or have become destabilized. And that was really fascinating to me, the talk you gave a couple of days ago on that.
Thank you.
Should I go down this list of questions or?
Do you have somebody in text to you or on the chat box just to you?
From the chat box. Okay, so the one that stands up right here is which method have I used to isolate EVs? I kind of went through that quickly. It's been published. It's a centrifugation method, low speed, middle speed, filtration through 2.22 micron. We have to concentrate, and these are all because of the toys that we have to our disposal. So we don't have the biggest centrifuge, so we have to concentrate now slow speed, middle speed, centrifuge, medium. So in theory, we're getting rid of so-called microvesicles. Then we filter that, we concentrate that, and then we use high-speed ultracentrifugation to pellet those things down. We've tested the various fractions in between these, by the way, so before you jump all over me about these aren't true vesicles. The other pieces don't show these activities. We've also taken the medium, or the high-speed supernatant, which would have so-called exomers potentially in that. That doesn't have the effect either. So it is what it is, and we can argue about how well that's done or how poorly that's done, but it gives us stuff that's active. It's different between different tumor types and definitely between different so-called normal cell types versus tumor types, and the fractions in between don't seem to carry that same impact.
Mike, I have one. You covered a lot of pathways, and then you mentioned this drug early on. So can you comment on that? Is that drug acting through any of these pathways, or anything known about its mechanism of action?
Which drug? The ERK inhibitor?
No, you mentioned the one drug for therapy for glioblastoma, or?
Temozolomide. Is that the one you're talking about? That's the standard of care. So, like pretty much everything in the brain tumor world, we find out what people do in melanoma first, and then we chase it down. So in treating melanoma metastases to the brain, they found that temozolomide could cross the blood-brain barrier. It had some effect. So that was co-opted by brain tumor people. They found you had to combine it with radiation. So what it's a DNA alkylator, and it's not a very good one at that when it comes right down to it. So it's not necessarily something that's going to directly impact a signaling pathway, but there's probably implications of that. What we did find was that if we treated tumor cells with their own vesicles, that would improve their resistance to temozolomide in terms of just growing in culture. So that was kind of disturbing to me, because tumor cells are likely spitting vesicles all over themselves all the time. So they sort of have a built-in defense mechanism at that level, and we didn't pursue it any further. And we're like, wow, look at that. But there it was.
You have a couple other questions. Did you use astrocytes from adult donors?
Everyone we used in culture, those were fetal, right? And this is a big question. This is kind of what we're stuck with, and I mean we can buy these, so that's the nice thing about that. The tumor chunks themselves, those are all adults. So whatever similarities we're seeing there, I would argue are perhaps related to just being astrocyte-like, but that is something to think about, that the astrocytes we're looking at might not be exactly the same as we'd find in an adult donor. But I would also point out that we've seen very similar biochemical impacts on pig brain. So this kind of crosses species at that level. So that's a great question. That's an issue that is hard to get around. Let's put it that way. EVs in the blood of GBM patients. We have done that to some extent. The arguments being that you've got a large gamish of stuff there. Mostly what we've seen on that is if we take those, and this is the only study I think I would say we've done in any depth, we put the so-called plasma EVs purified from a brain tumor patient, glioma patient, put them on the brain tumor or on the brain slice, took the media from the slice, and found that it still had significant gelatinase activity, as did the vesicles that were in the blood itself. So there'd be an enhanced chewing ability, shall we say, of the plasma vesicles. We've also done that in the T cell work, where in fact, we used the same patients. We were able to use the same from the cell line as well as from the patient's blood. We were able to see that those indeed had detrimental impacts on the T cells themselves. In other words, activated T cells become deactivated. We saw pretty much the same story.
Mike, there's one other question in the chat right above the one you mentioned. It says, do you expect similar effect of GBM exosomes on other cell types, neurons, parasites, endothelial cells, etc.?
Not necessarily the same effects, because these are very different cells that would respond to it. Some of this work has been done by Zandra Brakefield, for instance. She's kind of allowed us to continue working in the astrocyte space while she has pursued the microglial space. And what they find in the microglia is these are essentially the macrophage, so to speak, of the brain. And those microglia will become essentially, once again, supportive of the tumor. They will become immune suppressive. They will emit molecules that make the tumors grow faster. So a very similar, the general concept of the tumor dealing with other cells makes the other cells do things for the tumor. That seems to be the common pattern here. So consequently, they're not going to be the exact same story, but you're going to see, I think, very similar big overall responses. From the endothelial cell story, there's some work going on with that. These tend to become what they call a senescent phenotype. So it's interesting how those are changed in ways that are not necessarily the same as an activation phenotype. And then the neurons, there's a lot of work going on with that I'm not even feeling equipped to discuss, but that's a great question.
Okay, great. Well, if no further questions, Mike, thank you very much. Very interesting talk. And this will be, I think, available, posted on the portal at some point, the recording. So thanks to all of you for attending, and we'll see you next month. Thank you.
Okay, thanks.